Good evening. Feels odd to say good evening when it's still bright and light outside. I love summer. Nevertheless, it's officially evening. My name is Leslie Gordon, and welcome to the U.S. Geological Survey in another installment of our continuing public lecture series. Because I want you to come back next month, I want to let you know that our talk on July 28th is Dr. Margaret Mangan, who is the scientist in charge of the California Volcano Observatory. A lot of people forget that California is not just earthquake country. We have several active volcanoes in the state. So do join us next month to listen about the California Volcano Observatory and the work we do in monitoring those California volcanoes. Tonight's talk is Lisamarie Windham-Myers. She is a research ecologist in our Water Missionary National Research Program. Excuse me. Lisa is an ecosystem ecologist specializing in wetlands. She received her bachelor's degree at UCLA and her master's and PhD at Rutgers University in geography and ecology. As an ecosystems ecologist, she works across a broad spectrum of wetland types on issues related to carbon, nutrient, and mercury cycling. And although Lisa is interested in the relative importance of hydrologic, soil, and plant-driven processes across sites and seasons, and how they can be used to predict or manage environmental conditions. While her focus on mercury began in New Jersey meadowlands, Lisa is an active researcher in the biogeochemistry on all U.S. coasts, and for the past 12 years has played a leading role in providing science advice to agencies seeking to reduce methylmercury exposure here in San Francisco Bay. So please welcome Lisamarie Windham-Myers as she talks about mercury and rice in the California Delta.
Thank you. It's nice to see this crowd on a beautiful summer evening. I am going to try to tell you a story about the interaction of these concepts of mercury and rice, starting with the more global context for mercury and global context for rice, bring it down to a very specific study we did in the Yolo bypass, which involved a whole lot of interacting processes, and then take it to what that means for the California Delta as a whole. So as you can imagine, with that kind of scope, you have a really broad set of partners, and we do. There's many agencies and institutions that we've been working with for the last 10 years. And these are different sites, different questions, different focuses, but they all tie into what I'm going to present today. And it's not really just about the agencies and that are providing funding in the institutions, all those providing scientific support. It's about the people. We have a lot of dedicated people that ask these kinds of questions. And I'm just doing a shout out here to the USGS folks that I work with most commonly. Marvin D. Pasquale is in the upper left corner, and he's the lab manager for a world-class mercury lab that we have here. I wouldn't be here without him or this team, which includes Jennifer Agee, Lee Q, Evangelos Koukouros, and Michelle Arias, and also Melissa Moradian is not on here, but she's our newest member. And then we also work with the California Water Science Center, which is in Sacramento, and the Western Ecological Research Center, which has offices in Vallejo and Dixon, as well as other places. And some of the people there would be Charlie Alpers, Josh Ackerman, and Jacob Fleck. So when we talk about mercury, most people think of it as a local San Francisco Bay story. We have a lot of contamination, but it's really a global pollutant. We actually have a 2013 treaty. There's a Minamata Convention that was passed, 128 signatories for countries to reduce their emissions of mercury. While there's no biological purpose to mercury that's ever been shown, the use of mercury in all kinds of things like light bulbs, switches, batteries, thermometers, this has been known for a long time, and it's because mercury has a really low boiling point. It can be in a vapor form in environmental, like room temperature, basically. And so it's a very special metal, and that's why emissions are a concern. it turns mercury into a non-point source. So when mercury gets emitted, it can be in basically three main forms. There's Hg0, which is elemental mercury, and that's the one that's really fairly inert. It goes up into the atmosphere, stays there for a while, makes its way up to the poles. This is what's getting up into the polar bears. And that's the long distance one, the global transport one. Mercury 2+, or divalent, or sometimes called reactive mercury, is basically a sticky gas. So when mercury gets oxidized, it can glom onto things. So it'll glom onto raindrops or particles, aerosols, and then it can deposit out from the atmosphere. So it has a less long distance transport. And then the third is particulate mercury, and that's what you find in smoke. So like particles that you might see in a coal burning power plant here. And that has a really short transport. That's basically just gravity. It's going to move until it falls out of the sky. So we look at all three of those and, at a global scale. But when you look at emissions and what's really important, these two really pop out. So coal is the second most important current emissions of mercury. And that's mostly coming from China. That's the current sort of big source of mercury. But the biggest source is actually artisanal gold mining. And we've discovered this basically through remote sensing. Whenever the price of gold goes up, people go out into the jungles of the Amazon, into Africa, into wherever, and start just trying to pick up flecks of gold. They bring mercury with them, and I just think it's me with a little feedback, or are we okay, good. And so we can see basically this grow. Whenever again the price of gold grows, we see more and more artisanal mining, and the current rates of emissions are about 700 metric tons per year through that process. Actually, just as a note, Peru just put out a state of emergency in 11 jungles for mercury contamination to peoples there. So it's a definite health hazard as well as just entering the global system. But before we sort of wag our finger at these people who are going out with mercury and clicking up gold, that was us 150 years ago. We did the artisanal mining up in the Sierras. And the reason we were so successful in the gold rush is we had really quick, cheap proximity to mercury, because the mercury is here in the coast range. Right under our feet are mercury ores that can be used to get mercury out of and then carry that mercury up into the gold-bearing sediments of the Sierra. There's a fact sheet by Charlie Alpers from 2005 that I can't go into all the details of it, but if you're interested really in the history of mining and how mercury has been moved around, that's your source. I would go there. But just to talk about what we have right here, again, mercury ore that's beneath our feet is mostly cinnabar. It's a mercuric sulfide. It is really stable. You aren't really going to get mercury out of that ore. You have to roast it, and so you have to bake it at a very high temperature. And then you can volatilize the mercury out of that rock. And here's a photo from New Almaden. Once you've volatilized it out, you can then condense it and collect the quicksilver or the elemental mercury that's running out of this condenser, and then bottle it up and take it wherever you want to. Just to put in perspective the amount of mercury that came out of New Almaden alone, the values are huge. From this one place alone, 7,000 metric tons were released in a decade at the height of the mining period, and that's basically about 700 metric tons. It's the current emissions of the world right now globally. So it had a very pulse period. We stopped mining it basically once it got to be around 1920 or so, not so much. Most of it had been used and then already brought up to the mountains by that point. That said, 99% of what was brought up there is still up there. It hasn't all run out. It is still up there. And so if you go out with a pan or kind of dig around in some rocks or some sand, if you're lucky, you might actually see a little blob of elemental mercury out there. It's not that hard to see. And yeah, the concern is that basically what happened in the Sierras is not staying in the Sierras. It is coming out in a pulsed fashion, but eventually it's all making its way down into San Francisco Bay. And we've got that mining the gold mining based mercury coming out from the Sierras, and then from the coast range, we've got this roasting leftover mercury coming out through, for example, Guadalupe River there. And so the whole bay is basically pickled in mercury. It's just out there in the sediments. There's just nothing we can really do about it. So we actually use a term often it's called bay clean, and it basically means sediments that are sort of the base level, so anything less than about 400 nanograms per gram. That's really high, but that's our base level. And it's just the way it is. Getting rid of this, it's going to take 100 years for it all to get it's going to take far longer than that to get it all flushed out, for example, to the ocean. But it would be very disruptive to try to dig it out. And it's basically there. We also have striped bass concentrations that are fairly high. We have a lot of fish that are high, but we needed something to monitor through time. And so for the last 40 years, there's been regional monitoring programs that just measure striped bass. They're about a foot long is the standard size that we look at. And when you look at the concentrations, they tend to be high in many places, but particularly sort of in the edgy type environments of the Bay Delta. The value of 0.43 is actually really close to a can of white albacore tuna. So if you think about a striped bass about this size, that's about the concentration that you would find. And FDA actually recommends for pregnant women and children just 1 can a week. So that's about what we're talking about with these fish. Then of course, we don't even have numbers here for the monster fish, which is really what everybody's out there to catch. And the bigger the fish, the more biomagnification, the more concentration, the older the fish, of course, and there's more mercury in it. So we do have an issue with mercury in our fish. You might say, oh, it's not so bad, a can of albacore tuna. But the point here again is we're higher than everybody else. So if you go to other estuaries in the country, they tend to be lower. Anybody that's been monitoring striped bass will basically have lower average mercury concentrations than we do. And there are populations of folks in the Delta that basically have high concentrations or concerning concentrations in their hair. So we know there's a concern, a local population concern. And that's why you see these signs everywhere in the Delta, in the Bay Delta, telling you what fish you might want to eat and how many times a week and maybe what kind of fish you might want to avoid. But this puts a real wet blanket on the fishing and tourism industry for the Delta. It's a $2 billion industry, and it would be great if we didn't have this problem. The other big industry in the Delta is farming. I think we know that. Interestingly, at the same time we were putting all that mercury up into the atmosphere, we were also levying up the wetlands of the Delta. It used to be one huge freshwater wetland, and now here you can see in red that the majority of the Delta lands is actually farmed now, and that's because we built levees and then allowed them to be drained. And so we have very channelized rivers that are channels, I should say, in general, in the Delta that are great for boating, and then we have great farmland. And this industry of farming is huge. It's about $27 billion for California. 100 years ago, all of that red would have been green, would have been wetlands. It's just for clarity, it's a really large amount of wetlands that we have lost. So these Delta levees have just changed the whole function and appearance of the Delta. When you go out there, you either see the farming land or you see the open water, depending if you're on a boat or in a car, basically. And again, it's a very lucrative part of our state. And we have these signature crops out there, things like asparagus and pears. And increasingly, we're starting to see rice moving out there. And people like to put rice out there because it helps prevent peat oxidation. If you put rice out there, you can keep the water levels higher, and then you don't have as much of the loss of those peaty soils. They're not blowing off. So there's an interest in putting rice out there. Right now, rice is actually really important in California. From a global perspective, we're the 5th largest producer in the world. We produce a lot of rice, and more than 50% we export. And we grow really high quality rice, the kind that people pay top dollar for. So a lot of it goes to Japan. So the big barges come in with Sony TVs into Sacramento, dump their load off, and go back with rice. Pretty common. You can see that where the rice is mostly in the Sacramento Valley, and then there's some sort of popping into the delta. Probably the most important feature of rice for me is its role in as a surrogate wetland. Since we lost so many wetlands, today for every 1 acre of natural wetland, we have 2 acres of rice. So rice is by far the most common wetland type in California. So when we have a drought and our natural wetlands aren't getting wetted, but we're putting water on rice fields, that is where the wildlife goes. So for the last few years, there's been some really good documentation by the Nature Conservancy about where the birds are going, and much of them are going to these rice fields on the Sacramento River, because that's where the water is, that's where the wetland is. So they play this really important role in California as a wetland surrogate, and they are effectively an agricultural wetland. From a global perspective, rice fields are the most abundant type of wetland in temperate and tropical latitudes. They all have the exact same species. All rice fields have Oryza sativa. And that they have just bred for the last 4,000 years Oryza sativa to grow white rice, black rice, red rice, long grain, aromatic, short grain, medium, whatever. They're all the exact same species. And so if you think about the importance of that, the world is dominated by this one plant. These wetlands are dominated by this one plant, and it's a really productive plant. So it's incredible breeding that's gone on to make this plant. Half of the biomass in these plants is edible. So it's the grain. The grain is very weighty. So it actually makes sense to do things like these crazy terraces on these steep slopes, because you get so much food out of a very small area. And when we look at cropping across the globe, here in California, we basically harvest in October. The Mississippi Valley here harvests about September, and we have just 1 crop a year. But if you go out to these tropical wetlands, these areas, they have up to 3 crops a year. There's no problem. Growing a crop takes about 30 to 60 days. 60 days is when you usually max out. Then they pull it out and then put the next one in. So we grow a lot of rice around the world, and it's just a very important food source. So why do we care about rice and other wetlands? There's a plus side for studying this, and there's a negative side for studying this. So the plus side is, of course, food supply. We have a growing population. This is a really excellent plant for making lots of food fast. The negative side are public health issues. Mosquitoes, it's becoming a growing concern around the world. Methane, rice is currently responsible for 11% of the global methane emissions. It's a huge source. And thirdly is methylmercury. It's starting to pick up, and people are concerned about it, because when you look for it, you tend to find it. So wetlands, it is absolutely no surprise that wetlands make methylmercury. This has just been demonstrated over and over. At the micro scale, you can see it because the critters, the bacteria that actually do the methylation of mercury, adding a methyl group to a mercury molecule, they tend to be in wetland soils, any place that has lots of organic matter and anoxic conditions. That's where you find it. And we also see it at the macro scale. This is a map of methylmercury concentrations in surface water, and the best predictor of methylmercury concentrations in a watershed's surface water is the percent of wetlands that is in that watershed. Also, dissolved organic carbon is a good predictor of how much methylmercury is in the water, but that's also a wetland product, so it just plays this important role. This diagram is a lot of moving and shaking of mercury and methylmercury, and it being produced and decomposed and so on. But I want to show it just to emphasize why, of all anoxic soils, why wetlands are so good. And it's because they have this sort of suboxic zone. It's not too oxygenated, it's not too anoxic or anaerobic, it's just sort of right in the middle. And so you have the right conditions for active bacteria, but the mercury itself is not glommed onto a sulfide molecule. If you think about mercuric ore, that's a strong bond, mercury and sulfide. The more sulfide you have produced, the less mercury is available. So there's this sort of suboxic zone where sulfide is low, but you still have the active bacteria. That's the good thing. And we've learned now for the last 10 years or so that roots of wetland plants are really good at kind of building up that suboxic zone. They're leaking out oxygen, they're also leaking out carbon, so this is a really good hotspot for methylmercury to be formed. So to have a problem, though, in a wetland, it's not just based on production. It has to leave the system, leave the soil, and then enter a water column or such, but then ultimately get into the food chain. And bioaccumulation is mostly becomes a problem when you have biomagnification, when you have a long food chain. So the mercury just keeps getting more and more locked into tissues as it moves up the food chain. It's hard to get rid of once you've got it in your system. So what's interesting about rice is that it doesn't need the transport to get to bioaccumulation, if you will. The transport just happens in the plant itself. It's really good at moving methylmercury up its stems and just right into the rice grain itself. So people have been concerned about arsenic for a while in rice, and that tends to be in the brown part, the rice bran, the part that goes around the rice, what you call brown rice. And so you can actually polish that away, and then it's free of arsenic or somewhat cleaned. If you polish the grains to make white rice, you don't get rid of the methylmercury, though, because it's in that endosperm. So this has been starting to be an interesting issue, particularly in China, because they grow a lot of rice, they do a lot of coal combustion and other mining practices, and the population mostly eats rice as a main staple food, so it's actually the dominant way that some populations in China are picking up methylmercury. It's through their rice consumption, not their fish consumption. So if you look where all the research is being done, it's mostly being done in China, and the reason is they're trying to figure this out, and they're trying to come up with a solution. One potential solution is genetic varietals that aren't moving that methylmercury so quickly. Another is the idea of aerobic rice cropping, or basically keeping the soil not so wet that you're going to make methylmercury and that it doesn't move up into the plant grain itself. And that's turning out to be promising as well. So rice fields here in California, we've been studying them for a while. This is data from Mark Marvin Di Pasquale. There's over 1,000 data points in here, just summarized into a very, very simple diagram, which has basically methylmercury production on the bottom and percent methyl in the soil on the y-axis. And the key factor here is these are log scales, is that wetlands shown in the green are really the sites that are making methylmercury. These are where the high production is happening. It's not necessarily happening in the bay sediments or sloughs or lodic, meaning flowing streams. It's really happening in these wetland soils. And when we look at really across those systems, what is the big driver? It tends to be the abundance of organic matter and extreme wetting and drying. So not like a tidal system where you have, it's a little wet, a little dry, a little wet, but it doesn't ever really dry out. It's the sites that really dry out. Those are the ones that seem to be making the most methylmercury. And it might be related to resetting that sulfate iron clock, getting those to the right form to support the sulfate and iron reducing bacteria. So where are these wetlands? Well, the wetlands, I think I showed you on an earlier map, are mostly out in these hinterland areas. They're not in the central delta anywhere. They're out in the edges like the Yolo bypass, Cosumnes River, and then around to the south as well. And there's a pattern that's just been very persistent through time in fish, and it's called the hole in the donut. You'll hear this term a lot. Not donut hole, that's the part that comes out, but the hole in the donut. And if you were going to go fishing, this is where you want to catch your fish, because the fish tend to be cleaner right in that central zone. These data that are being shown are for silversides, which is often used as a sentinel species because the species doesn't leave its home very much. It doesn't range very far. So it has high site fidelity. And the fish concentrations tend to be, again, low there for this good sentinel species, and then higher in these outer areas. So a lot of people think, well, it's the wetlands that are responsible because that's where the methylmercury is being made. That's one of the possible explanations, but there's really four. And I just want to lay them all out because these are what people are trying to figure out right now because it very much affects how you manage the delta. The second possibility is just proximity to mining, that there's more mercury coming in on those tributaries that are coming in from the Sierras and from the Coast Range, the San Joaquin. There's also the possibility that the water comes in through these tributaries dirty. It has methylmercury already coming in, and then by the time it gets into the delta, where we have these more open water type environments, you can have settling of that particles that are in there, so the mercury drops to the bottom, or it gets what's called photodemethylated, or degraded. The idea is that sunlight hits the mercury, the methylmercury, and then it basically breaks up that bond of methyl to mercury, and then it just becomes mercury, and that's not so much of a problem. And then the fourth possible explanation is just pure hydrodynamics, that the central delta is a really well-flushed system. So it's just a matter of dilution. The water comes in and it just sort of gets cleaned out on a daily basis because it is a tidal system. The delta is still tidal. So I'm going to focus here on the methylmercury production issue and whether that's a possible explanation for these high concentrations, because that's what our focus has been. And we've been specifically working with the State Water Resources Control Board to get a handle on if there's any management practices that you could do to reduce the methylmercury concentration in the waters coming off this land. They're trying to implement the first ever total maximum daily load, or regulation for export, for methylmercury. Nobody's ever tried this before. They've done it for total mercury, but this is a species of mercury. It's really hard to measure, it's expensive, and it's always changing, so it's a very tough thing to pull off. But their argument is there's so much mercury out there, we really want to figure out the methylmercury load. That's the part we're concerned about. So again, we're trying to get from an average of 0.31 nanograms per liter in the Yola bypass down to 0.06. That's 80% reduction. Not sure if we're going to get there, but that's the goal. So talking about the Yola bypass, anybody here been out to the Yola bypass wildlife area? Very few people. Yeah, okay, maybe about 6. Yeah, it's just stunning, and particularly when they have things called duck days. They have some really good migratory waterfowl moments to catch. It's a really wonderful place right along Highway 80. And out there they have basically a wide variety of wetland types. We were focusing on agricultural wetlands, so rice, wild rice, which is zizania, which isn't a true rice, it's just a big grass, makes the black seeds, the wild rice you use at Thanksgiving. And then fallowed land where you basically grow out the weed crops over a couple years. And so that's the types of systems we're looking at. We looked at the wetting during the summer, and then the dry down, and then the wetting during the winter, and then again the dry down. So we looked at basically all these processes at those different time periods over a whole year. We also looked at a permanent wetland that was flooded all year, and we then looked at a seasonal wetland that was only flooded half a year. So when the winter rains come, then it was flooded. And this was work done with the Department of Fish and Wildlife, Yola Basin Foundation, Moss Landing Marine Labs. It was a very large group, and you can see why. We looked at many different components. I'm going to get my glass of water. Forgot. And I'm trying to lump them into classes like water, sediment, fish, and so on, but we were looking at processes. We wanted to understand how the mercury was moving through these different pools and how that changed over the course of the year. So lots of things were measured. This was about $1 million project, and there was 12 PIs, so we were very integrated, very big project. And the goal, again, was to try to link those processes in a box model to figure out how these systems work. So, can't be me. No more water, okay. So we measured basically kind of the obvious thing. We measured the methylmercury that was coming into the system and the methylmercury that was leaving the system. So just a load difference. How much was coming in, how much was going out. And the difference between those would be net export, the amount that was generated by the field, for example, what the difference between those two inputs and outputs. But we didn't just measure that. We measured that all the time because we just wanted to get a sense of how things were moving. But we also measured all of these internal processes of creation of methylmercury, degradation of methylmercury, moving into the soil, moving into the water, moving into the plants, moving into the fish, the whole full Monty here. And this ended up being a special issue of Science of the Total Environment, and really good papers. I would highly recommend, if you're interested in this, to go read the papers, but I'm going to just summarize briefly what we found. The probably most important thing was timing. We found that all of those processes were related if you step back and looked at how they were, but they was all lagged. They didn't always happen at the same time. So the summer turned out to be really important for methylmercury, the summer was not so, methylmercury was produced all year long. It didn't really matter if it was the summer or the winter. It was just being produced whenever there was water on the land. However, export was really low in the summer. It just was not leaving the system. And then lastly, biotic uptake was high in the summer. That's when we saw the fish really picking up lots of methylmercury on the fields. That study was done with caged fish, so we kind of locked them in place and saw how much methylmercury they picked up. It was a completely opposite pattern in winter, where export was high and then the fish uptake was low. So this is the type of patterns that people don't think about. They're important to choosing when you're going to monitor a site to understand actually how the system is working. Another thing we learned was that hydrology was really important. No real surprise here, but I think some things that were important was the timing and flooding led to hot moments. We would get sometimes, you put the water on, you get a big spike of methylmercury coming out of the soil. I'll show you some data on that. And so if you know when the water's going on, you can know, if you know when it's going on and off, you can know when you should be monitoring it to understand what the real net benefit, or sorry, the net export is going to be. Another thing important was source. So this had to do with basically dirty water or clean water. So if you had methylmercury in your water already going onto your land, the rice fields in some cases actually cleaned up the water. They provided some temporary sinks for that methylmercury, so it went out a little bit cleaner than it came in, And if the water came in on the cleaner side, it could leave dirtier because it had more to pick up along the way. So the type of water that went in sort of drove what was going on. And then finally, and this really stood out, was that plants were not just passive players in these fields. They're really dense. They're a huge physical presence out there, and they were controlling the hydrology, flow paths, movement through the plants, through transpiration. They were affecting carbon availability and therefore the production of methylmercury. And then they were also shading the landscape. So we had thought in summer we'd see a lot of photodegradation, a lot of sunlight hitting the water, and then it would break up the methylmercury. This would be great. But the plants basically shaded it by about 90%. So there was just very little sunlight actually reaching the water. So these are the kind of things we learned. So I'm just going to step through some of the data just to put you in perspective of this large study with lots of data points. This is the full year on the x-axis of data measurements, and on the y-axis we have unfiltered methylmercury in the water in terms of nanograms per liter. This is also another way of saying whole water, so it was just straight water, it wasn't filtered. And the first thing that you should notice is that every sample, it did not matter if it was the inflowing sample or the outflowing sample or a sample from the middle of the field, they were all high. So everything was above the target goal of 0.06 nanograms per liter. So this is a really tough goal to try to get down to 0.06 when your incoming water isn't even there. I also want to just orient you that, again, triangles are the inlets and circles are the outflows. And all I'm really going to talk about here is the outflow data because it's sort of the most important points I want to make. The hydro period, of course, wetted, then dried, then wetted, then dried. And when you look at the patterns that we saw, the first one that probably leaps out at you are these big screamers here for the wild rice, high concentrations in the output water of the wild rice fields. And those were the highest I think anybody had measured in the Delta period. And it seems to be related to the fact that they wet harvest. They basically leave the floods field, they beat on the wild rice, and then it floats kind of like cranberries, and then you collect all the rice, and then that's how we collect wild rice. It's different than white rice, which you dry down and then harvest. Another thing that popped out was with white rice, the big spikes that were different than the inflows, the big outflow spikes, tended to happen right on flood up. So as soon as you put water, it didn't matter if it was the summer flood up or the winter flood up, that's when this big pulse of methylmercury would come out of the system. And it looks like this is basically legacy mercury. It was methylmercury. It was made in the previous season, and it's just sort of sitting there in the dry sediment, then you put the water on and whoosh, the flush comes out. So it's not so much a active production at that time, but more of a physical release. And then the final important thing, and this is starting to become a general rule, is that the permanent wetlands were always low at the outlet. So the process, all these processes of production and degradation, they're always happening in all of these wetlands, but degradation seems to really be a bigger player in permanent wetlands. We have less methylmercury coming out from the permanently flooded wetlands than the seasonally flooded wetlands. So what happened when we looked at the fish, the bioaccumulation? We saw that even after 30 days of exposure, they were picking up a lot of methylmercury from the water column. So these are really high rates of bioaccumulation. These are little mosquito fish. They're really, really tolerant little buggers. They were surviving at like 40 degrees C water. And they basically, by day 60, the end of the experiment, they were 12 times higher than initial concentrations in the white rice fields, but only about 3 times higher in the permanent wetlands. So there was accumulation in permanent wetlands, but it wasn't that high, and it was almost in keeping with what you would expect. It was, the white rice was just a really high accumulation in the fish. And just from that perspective, these are well above the FDA guidelines of a fish that you should eat, and these are little tiny mosquito fish, so they're very base of the food web. Anything eating them is just going to get more and more mercury in them. So one thought of why the methylmercury would be so high in the white rice fields was that it was the fields were making so much methylmercury. There was a big source there. And so we looked at production in many ways to look to see if it was just really kicking, and that's what we thought. We thought we'd see really high methylmercury production, and we did. It was reasonably high. But we wanted to figure out why it was high or not, so we looked at the 2 main factors that drive methylmercury production. One is the availability of reactive mercury, or that bioavailable pool that can get methylated, and then the other is the bacterial activity. So we looked at these 2 in concert to see which one of those were driving the story. Another way of drawing this is what's called often a Goldilocks model. The idea that there's this just right condition, and when you're in this zone, you have high methylmercury production, but when you have lower rates of sulfate reduction or more sulfide in the soil, you have less methylmercury production, because again, you have something, the teeter-totter is pushing on one direction or another. So I'm not going to go through the data in extreme detail, but I do want to just say that we did not see a difference between the different wetlands. They all effectively made methylmercury at the same rate. So it wasn't the methylmercury production rate that was driving the differences between the fields, and the wetlands in general. They were similar, but for totally different reasons. There was a lot of reactive mercury available in the rice soils, but they didn't have as high bacterial activity, actually. Whereas the managed wetland had very low mercury available in the soils, and then much higher bacterial activity. So for different reasons, they were both sort of in the moderate range. That to say, they were not low compared to other places in the country, but for this system, they were lower than we thought they were going to be. So what explains these differences that we saw basically through time? We then basically started looking at physical processes. We're like, well, what's happening? How is this stuff moving around? And it turned out to be a really interesting story with the plants. Transpiration turned out to be 90% of how water was moving through these fields in the summer, so it's shown here in the black. And when people think about ET, evapotranspiration, they just think of it as one lump term, that this is how water leaves the system and moves into the atmosphere. But they're very different processes. Evaporation leads to concentration of constituents in the water, whereas transpiration is pulling that water down into the soil, and then water's leaving through the plants, and so you're getting a concentration of any constituent in the soil. So where the mercury is completely different, depending on which one of those is the driver. And so you can see here in the summer, transpiration is a huge driver in these fields. You can just think about what this, it looks like a lawn. It's just completely dense, 2,000 grams of carbon per meter squared, 2,000 grams of biomass per meter squared. It's a huge amount of material here. And then in winter, again, very little biomass on the field, so therefore not a lot of transpiration. So what that really looks like is that just from a pure physical process, something as inert as chloride, we use that as often as a conservative tracer, would be pulled down into the soil during the summer, and then released in the winter. And that's exactly what we saw with methylmercury. It's being pulled down in the summer and then released in the winter. And that's happening, this is leading to a limitation on methylmercury export in rice fields for two reasons. One, it's physically pulling water out of the surface into the soil, so some of that's bringing some methylmercury with it. And then the second is that a vective pull down prevents the diffusion up of a higher concentration that's in the soil. So the diffusion can't compete with the strong advective pressure. So there's a very strong physical story there. And then, as I mentioned again about shading, just to put this in perspective, the rice field looks like this in May when you get started, before the plant starts growing. And then by August, this is white rice. You can have this hugely dense canopy of shading, and it basically made the amount of sunlight hitting the water be equal between the winter and the summer. The summer, even though there was, on average on a daily flux, it was at 50 moles per meter squared per day, by the time you factored in the plant absorption of that light, only 5 moles were hitting the water, which is about what you find in the wintertime. So another plant-related part of this was we did what we call devegetation experiments. We didn't let the rice grow in certain areas. We did a lot of replicate plots where we said, let's just keep the rice out and see what happens if we don't have rice in that system. And in those devegetation experiments, we measured tons of things in the soil. And what we found is that basically, yes, if you did not have plants there, you could reduce methylmercury production by about 60%. So you want to shut down methylmercury production and get rid of your plants. And that was mostly being driven by a reduction in the bacterial activity in these sites. In particular, we looked at things like sulfate reduction, also depressed, and what we saw was, again, it was pore water acetate as a carbon source. I'll go there. As a carbon source, that was really what was driven down. So the plants were bringing carbon to the system. When you took out the plants, they had less carbon. That looks like why the bacteria were less active. We definitely saw less sediment methylmercury in those sites. We know that the proof is in the pudding. There was less methylmercury in the soil. And then finally, we measured other things such as pore water chloride as a conservative tracer and iron speciation. And what we found is some evidence, again, of this rhizo concentration, so again, concentrating in the soils, and then also rhizo oxidation, basically keeping what was sometimes called the Ferris wheel going, where the iron is basically moving from ferric to ferrous, or from 3 to 2, and we get more moving back into 3. The more oxidized form, we get that happening in the vegetated sites. So the data I just showed you is basically here plotted in green in a more distributed function here. So we have pore water acetate, again, log scale on the bottom, and then we have the bacterial production rate on the y-axis. And you can see that basically more carbon in the soil, more bacterial activity, definitely true for the summer. And then interestingly, it's true for the winter as well. So we actually had the highest rates of methylation were actually in the winter. So this decaying rice straw, we believe, is part of the carbon supply in the winter that's making these fields such good producers of such good producers of methylmercury in the winter as well as the summer. And then finally, we looked at the actual concentrations in the seeds just to see what was in there. We were mostly looking at it from a wildlife perspective. Are they picking up a lot of mercury by eating these rice and wild rice grains? And the answer was yes. There's more in the grains of rice and wild rice than in the cattails and tule seeds, which are also base of food web in some of these systems. So these values, they're higher than the native seeds, but just to put this in perspective, they're way lower than the median values coming out of China of this district that's getting a lot of attention and making concerns about human consumption. And it's basically more than 100 times less than you would find, for example, in a striped bass about this size. So they're fairly low concentrations, but they still were higher than what you would find in a native population. So that was the Yola Bypass Wildlife Area. And we learned a lot there, because that kind of study is very hard to replicate, all those moving pieces. But we've done some studies now in the Cosumnes River Preserve, and then also on Twitchell Island, which is right out in the central delta, both places where they're growing rice. In the Cosumnes River Preserve, they're growing it in organic fashion. They're adding less sulfate, for example. Sulfate is a really common carrier anion for ammonium sulfate, zinc sulfate, copper sulfate, whatever it is you want to add to your crop, they often tend to have sulfate attached. Not so much in the Cosumnes River Preserve. And then Twitchell Island, they're also growing rice out there, but this is a tough place to grow rice because it's actually pretty cold in the delta compared to, for example, the Sacramento Valley at night. And so we've just been looking at some key features there, and one of them is loads. We've been looking at how much is coming in and how much is going out from the rice fields there, and what we're finding is the same basic pattern. All of those rice fields are holding their mercury in the summer. They're producing it, but they're not letting it out, and then in the winter you get a flush out. And so they're basically sources in the winter, but not in the summer. We've been partnering with UC Davis on work up in the Sacramento Valley, because this is really where big rice is. The big fields are up there, and they have much less mercury in the soil. So it's really a question of, how important are they for export from that down the tributary of the Sacramento to the delta? There's a lot of the land, but there's maybe not that much mercury. But there's a graduate student named Christy Tanner who's just finishing up. There's wonderful studies that she's been doing. And this is just a paper that she just submitted where she's looking at the role of rice agricultural drainages on the concentrations of mercury in the Sacramento River at the point of the drainages and then also downstream. And she's saying basically the same thing. The concentrations in her system aren't as high as what we saw in the Yola bypass, but it's basically a story that the winter is important and the summer is not so much. So she's seeing a significant effect of the agricultural drains in the winter, but not in the summer. So this is all fine and dandy, and we can say that we have these general patterns. We understand when the export is coming, when it's not coming, but it doesn't really get us to the point of fixing it. So the real question that we've been interested in is, can we do anything about it? Now that we know all these little knobs that might be able to turn, could we actually reduce the amount of methylmercury being produced, and then how much is coming out of the system? So we've been at the Cosumnes River Preserve. We have a really good partner there in the Bureau of Land Management. They let us work with the farmers and do work on some of their seasonal wetlands. And we basically paid the farmers 3 years ago to treat their rice straw differently. We said, okay, either leave it on the surface like you usually do and flood it, till it into the soil, which some people do, and then thirdly, bale it up. Just cut it up, get it off the land. Let's just see if we can get a difference there. And we were really hoping that we would see a big effect, but we didn't. It was not a slam dunk. There was a lot of background variability between these fields, so we didn't get a really strong signal of that carbon removal. And so there's promising aspects to it, but it's the winter removal was not the real slam dunk we were hoping for. Another thing that we're trying up there is basically digging out, if you will, sort of a settling pond at the end of these seasonal wetlands. You could do this in a rice field as well. The idea is that the water coming off these shallow seasonal wetlands is loaded with methylmercury, and then if you give it a big deep area to go through, the methylmercury can settle out as particles if it gloms onto particles, or it can get photo-demethylated in that open water column and then come out cleaner. And this is actually looking to be somewhat promising. So where this is going is positive right now. We're seeing the same type of results in a Yola bypass study being done by Moss Landing Marine Lab. So this, I would stay tuned. I think it's gonna pan out to be a valuable approach. So basically, to be concluding, I just want to say that we've learned a lot thus far about how seasonal wetlands and rice operate. Rice is a really good surrogate for seasonal wetland because in seasonal wetland you have to you never know when the water is going to come or how much is going to come in. Rice is you know the day, you know you know exactly what's going to happen, so you can study it very, very well. And so we feel like what we've learned in rice actually applies to a lot of seasonal wetlands. The first thing I want to point out is that you really need to study the whole system. If you treat it like a black box, import and export, you'll get a story, but you won't really know why. You won't know why a site, for example, had production of methylmercury or a loss term. And that's what we're trying to tweak. That's what we're trying to understand. Some key points are that box models that we use today tend to be very big scale, annual averages. And in these pulsed systems where you have water coming on at different times, the wetlands in particular, but the delta too is a pulsed system. You need timing. You need to know when it's coming and what you're measuring at what point in time. The reason being a lot of these processes are concentration dependent, and those concentrations are changing through time, as well as the drivers of those processes. We found out that rice-dominated wetlands make methylmercury all year. They transport it mostly in the winter, and the real exposure to wildlife is in the summer. And that's not good for the wildlife because that's their sensitive breeding time, so this is not very good for wetland wildlife to have so much methylmercury on the landscape. We saw that as you increase the amount of mercury in the system, mercury pollution, you did see more methylmercury in the grains. The plants we saw were critical players in the system. So physically, in terms of controlling transport and shading, geochemically, the carbon exudation, the oxygen exudation, and then biologically, some of them accumulated mercury at mercury and methylmercury at different rates. And the optimistic side of this is we do think actually mitigation is possible. We're seeing that there are some knobs that you can turn, and there's some wetlands that are better than others, and we think we know why. And that's leading us to think that we can do some wetland management to reduce methylmercury export. That said, will this wetland management actually help reduce delta mercury fish levels? And I have to conclude that the solution is not in this piece of chalk. The solution is not in this talk. We don't know. We know that that's one possible reason. And what we're trying to do now is use our better modeling and monitoring tools to pull these pieces together. I'm just going to plug two real new advantages we've moved for. I'm going to show this video in a second, but on the right-hand side is some data from Sassoon Bay. This is the first map of methylmercury in surface water that anybody has made, and we just did this basically with a new airborne instrument out of NASA's JPL. This is from last year, and the bottom line is because methylmercury tends to track dissolved organic carbon, which is basically colored, the brown colored stuff in water. When you see like tea colored water, that's DOC or dissolved organic carbon, because you can see that from space or you can see that from an airplane. We found that there's a strong relationship, and you could actually map it. And this might be a monitoring tool that we can use to figure out where it's going at what points in time, where the concentrations are high. The model that I want to show you is something called Cascade. This is the website for, oops, sorry, I shouldn't have done that. The website for Cascade, which means computational assessments of scenarios of change for the delta ecosystem, is fascinating. It's a really great model that lets you link a lot of processes together. And this is just a physical transport model. I just want to show you basically how fast the Frank's tract here. Actually, I was going to make you guess where Frank's tract is, but I will just show you. It's the little red spot there. If you recall, that's a failed delta island that, of course, subsided. It flooded about 30 years ago. So now it's a big lake. So we just have a physical transport model on here to show where that methylmercury might move if it was in there and it wasn't going through any processes. So you can see the tides coming in and out and in and out. And twice a day we get a tide. And in this case, we have a summer situation, and we've got the pumps on. And what you can see is that even with very low tidal flushing, but with these pumps on, that Frank's tract gets cleaned out in less than 5 days. So anything produced in that site, it's out of there very quickly. In fact, it's on its way to Los Angeles. So that's the kind of thing that we need to do to figure out if a management process is going to have an effect at this large delta scale. And this is the type of stuff we're trying to push forward on. And just in case you're having rice for dinner tonight, I just want to point out that these are the kind of data that we have. If you look across the world, we are the only people doing anything ecosystem level in rice fields. The kind of data that's out there is pretty much only grain data. What's the methylmercury or mercury concentration in a grain of rice? And I just added these new points. These are coming in since we published this paper. We've gotten a couple new data points here. But YOLO bypass stands out, for example, for total mercury in rice grain. But it's the methylmercury that you really should be concerned about, and that's actually pretty low. So we're not having lots of high concentrations here, but in China is really where these hotspots of methylmercury are showing up in the rice grains. And rice is expanding across the globe right now, and there's global models people are trying to put together. If the Minamata Convention actually kicks in and we reduce emissions, would we expect to see an effect on rice grains in these new developing paddies around the world? And pulling that all together is difficult because we all know mercury is a legacy pollutant. Just because it was emitted, we stop emissions doesn't mean it's not still in the soil. So pulling these pieces together to understand how changes in mercury deposition and changes in rice locations might affect grain concentrations. That's what we're working with folks now, just basically sharing our data because very few people do this kind of ecosystem level work. And then finally, I just want to plug the California Rice Commission website. They have been documenting the wildlife visitation there for the last 2 winters, particularly during the droughts, and it's just amazing photos of really high professional quality photos of wildlife, in case you're interested. And with that, I'll just take any questions if you have any interest or anything you want to discuss any further.
If you don't mind, we have a couple of microphones in the aisles, and we'd like you to use the microphones for questions, because not only so that people in the room can hear you, but we record and stream this over the internet. And so if you're not able to, I'll bring you a microphone, but please step up and use that, and we'll just go back and forth between the 2 mics. And let this gentleman in the plaid shirt go first.
So 2 basic questions. One, what was the mercury used in mining? Just curious. What was the purpose of the mercury in gold mining?
It amalgamates with gold really well. So
Cool.
if you have little tiny flecks of gold, it's hard to pick that out.
Sure.
So throw some mercury on, gloms on, burn it.
Perfect. Okay. Second question. What's the effect of mercury on the human body? Meaning, I think it has a brain effect, but what is the actual effect?
Well, it sticks onto proteins. And so if you're growing neural systems, growing your brain and so on, if you're a pregnant woman or a child, it's going to be a problem because it's going to affect those developmental things. For us, it actually moves through your body pretty fast. It's about 50-day residence time. So like if you never ate fish and then you suddenly had some really hot fish, it'll show up in your hair like, that day, and then maybe a couple months later it might move down to here. So, it's not your body processes it and gets it out. It's when you're developing that it's really a problem.
Okay.
So,
Thank you. Appreciate
no problem.
Mercury is evil, but there are all sorts of other nasty stuff associated with agriculture.
Yeah.
Where does mercury fit on the sort of scale of competing with the other stuff for evilness?
Wow. Yeah, I wouldn't. Yeah, the axis of evil. All these things. I don't know if I could actually speak to that. I will say that it's the most regulated contaminant in the bay, but another big contaminant in the bay is PCBs. So depending on which hat you wear, you might say PCBs is a bigger fish concern or mercury. With agriculture, I'm not sure which are the real concerning. Like you're thinking pyrethroids or some sort actually, I'm wondering what you're thinking of bad.
Yeah, I'm not sure what the opportunities are here.
Yeah.
Did the rice people use a lot of insecticide
So not so much now because the market, we're going for a higher end market, I think, that doesn't really want the contaminants or the pollutants from that. But that said, copper sulfate is what you use to kill algae. and zinc sulfate is what you use to kill fungi. So there's, those are two things that are still being added in these fields. But they don't tend to accumulate in the rice in a way of concern, I think. So I think the bigger issues for agriculture in terms of toxics is organic pesticides, things like that. And I don't know how that would fit into a rice story.
Thanks.
Yeah, no worries.
Oh, so many questions. What's the original source of concentration in coal to begin with? And second, do we see any pattern geographically or in geological time in coal deposits? Do they differ wildly? And is Paleozoic coal worse than Mesozoic coal or such? And finally, what's the mechanism of biodegradation in the natural wetlands? Why is it so much faster there than in the rice fields?
So two things. Regarding coal, that there is a fascinating level of research on that, and I don't know if there's a paleo story, but I do know that we now can look at natural abundance isotopes, and you can know, if you look at mercury in the atmosphere, you can know whether it came from coal from China or coal from South America or coal from the United States. I mean, you can actually trace this stuff now because of isotopes. So it's, maybe there's a story there too for age. How it got there, most of these fossil fuel deposits are basically, old wetlands or old algal accumulation spots. There's, where old organic matter accumulates, and so metals are really common. Metals are really common in coal.
Yeah, I would think that if that's the mechanism, that then deposits would vary very greatly depending on geography and depositional environment, and so there's much cleaner coal, much worse coal.
Yeah.
What's the degradation mechanism for
So-
methylmercury?
So we think it's biological because it doesn't look like it's photo in those permanent wetlands, but it could be. A lot of the permanent wetlands have a lot of open water, and it hasn't really been teased out. But bacteria both methylate and demethylate. So that's happening always in the bacteria, and particularly more more aerobic type of species tend to do more demethylation. So pointing to where we don't have all the data in on where, how it's happening, but demethylation in the biotic or by the bacteria is important. I just don't have the.
And that would seem to open the door to bioremediation through your bacteria selection.
Yeah, but a lot of it's really the environment. So I don't know that you need to populate a new species to do the job. Maybe there's some that are more efficient than others, but that's actually on the table. To be honest, that is on the table as a possible. There's a lot of ideas out there using coagulant to glom up the methylmercury. There's a lot of ideas about how to clean these systems. And I have heard that thought come up, but I don't think anybody's tried it yet because the environment is so critical. We just don't have the right environments in most places.
I'm sort of curious as to what the industrial process was that led to the Minamata syndrome in Japan, and what ultimate. I suppose, as you mentioned, mercury is used a lot in the electronics industry. What do they do to mitigate the outflow of mercury from their activities?
Well, so you know the original Minamata story, just from earlier in the century.
Oh, well.
Okay, and just briefly, it's just kind of the worst mercury exposure experience that we've had is the lot from these basically processing plants. They were just running the mercury out into the bay, and it was getting into the fish, and they figured it out because the cats were going crazy because they were eating the fish, and then finally they figured out it was this mercury was getting into. It was a terrible story. But actually, I think there's a summary of that. There's a previous talk to mine, maybe a year or two ago, that actually spent some time discussing what happened in Minamata. So I probably refer to that one online because I don't think I know
Okay,
it.
all right,
the
I'll
details.
take a look. Okay,
Yeah,
okay, thank you.
yeah, thanks.
I have a quick question about the term methylmercury. You're using it interchangeable, mercury in the body, methylmercury. Is it processed the same way by the body? And what is the really dangerous? Is the methylmercury dangerous, or is elemental mercury dangerous? to the
Well,
body?
they're both bad, but methylmercury is just much, much worse. It's a much stronger. It's a methylmercury is just a mercury that has a methyl group attached to it, which allows it to be basically soluble, crosses more boundaries. That's what it gets glommed onto your proteins and causes problems. Mercury, frankly, I mean, it's not good. But it'll pass through you more quickly than methylmercury. So that's why people are concerned about that element, and that's why fish is the dominant pathway for us to be exposed, not, for example, light bulbs and so on. Oh, thank you. Thank you so much.
Well, thank you very much, Lisa, and thank all of you for coming, and I hope to see you next month.